Hello everyone, welcome to your lecture series on overview and integration of cellular metabolism. The first two lectures were on very fundamental topics. Today we will be starting with the metabolism proper and we will be going into carbohydrate metabolism, and our lecture will start with digestion and absorption of carbohydrates, where the concept that will be covered is how carbohydrates are digested, how glucose is absorbed, and how glucose transporters play a key role in absorption of glucose. So, very basic thing to understand is the digestion of carbohydrates actually starts outside your body only. How? By cooking. See, carbohydrate are actually made of units of glucose, right? Of course, glucose is a carbohydrate, is very fundamental, it does not need digestion. Digestion means breaking of a complex molecule into simpler forms so that we can absorb the whole thing, right. So, these carbohydrate molecules are connected by something that are known as glycosidic bonds. If we break this glycosidic bond, we can break carbohydrate. So, cooking is one such phenomena where the heat and mainly the heat actually helps in pre-digesting your food even before you take it. Okay? So, it is easier to digest a cooked food compared to a raw food, because our biological system with evolution has adapted in such a way that we can easily digest a cooked food. But wild animals, for example, a tiger can easily digest a raw food because there are mechanisms by which they can more efficiently digest any other raw material, especially carbohydrate. Okay. Now, carbohydrates, where they are found, it can be found in diet as both polysaccharide and disaccharide. Polysaccharide means multiple single units, multiple units of monosaccharides are connected. Examples are starch and glycogen. And disaccharide means two monosaccharide unit is connected with the help of a single glycosidic bond. Example of disaccharide is sucrose and lactose, right. So, what happens in GI tract, water breaks these down into monosaccharide unit. As we go down, the entire, whether be it polysaccharide or be it disaccharide, it will be easily broken down as it goes down into our gastrointestinal tract or GI tract, and we can then easily absorb them. So, the digestion starts with alpha amylase. So, any enzyme that digest glycosidic bond or carbohydrates are known as amylase, right. So, alpha amylase right at the buccal cavity, because it is present in mouth, okay, it helps in digestion, right. However, one very important thing to notice, gastric HCl, you might be aware that our stomach, the pH is very low because of presence of gastric acid. This inhibits salivary amylase. What is the importance? Importance is when the food is in our mouth and we are chewing it a lot, the alpha amylase is already digesting, because once we deglute it, it goes into the stomach, the action of alpha amylase will be inhibited. Therefore, right from our very childhood, our parents and grandparents used to say, please chew your food very carefully and don't swallow the food just like that. We are all in a hurry, we all just take gulps and we swallow, and then we go out and we frequently find ourselves with indigestion or bloating. This is the reason. Okay, so very fundamental but very important to know. So, in stomach carbohydrate digestion does not happen. So, it continues in pancreas, where the pancreatic juice has got alpha amylase, where it breaks down the alpha 1,4 glycosidic linkage. Again, what is alpha 1,4? When we consider a polysaccharide or multiple carbohydrate units, they look like this. There may be branching, they look like branch of twigs. Whenever we are considering this straight bond, these bond, they are alpha 1,4, these are 1,4, and whenever a bond is at a right angle, that is a branch point, this is alpha 1,6. So, alpha amylase can easily digest or break down this alpha 1,4 glycosidic linkages. So, if it works on this compound, it will continue to break till it reaches here. So, this ultimately one molecule will be left behind and the rest thing can be digested, right, you get my point. So, whenever it can reach up to the branch, and then we need more enzymes, okay, and that will of course be discussed when we are discussing about glycogen phosphorylation and breakdown. Everything will be very clear, but for now, to understand the fundamental concept of digestion, you should know that alpha amylase digests or breaks down alpha 1,4 glycosidic linkages, which will lead to production of compounds like maltose, isomaltose, dextrin. It may be branched or unbranched oligosaccharides depending on how complex our food was. Then the specific enzymes that are present in the intestinal brush bordered cells or succus entericus in small intestine, that is sucrase, maltase, isomaltase and lactase, they will act on those disaccharides, that will be converted to monosaccharides and then they will be absorbed. All right. So, this is an overview how the carbohydrates are digested. So, this is the thing that I drew in the previous slide, right. So, glucose unit, these are individual units of glucose that are connected by alpha 1,4 or alpha 1,6 linkages. So, this is a big starch molecule. For example, starch is a polysaccharide that we get in potato or rice, right, plant source. So, this can be broken down. So, for example, alpha amylase can only break it down till the branching point. So, this with few more than 2 or 3 units of glucose, when it is connected, it is known as oligosaccharide. Okay. So, this oligosaccharide can be converted to disaccharide, or this alpha amylase can directly break these two bonds and can be converted to disaccharide. Now, the enzymes in the intestinal brush bordered cells will again act on this disaccharide and then it will be converted to monosaccharide and it will be digested. So, the question is, if everything is digested, what remains? Of course, it is not digested, right. I told you in the intestine, it can only reach up to a branching point, and of course, something will always be there which is undigested, and this undigested part is actually the fiber component of our diet, which actually contributes to the stool volume, and this is actually good for our diet. Dietary fibers are essential for our diet. The role of dietary fibers will be explained in detail in later lectures, but for now this picture illustrates the whole thing. The digestion starts in the saliva, it does not happen in the stomach, okay, in the pH, no. Next, pancreas, from pancreas amylase is secreted, which acts on small intestine, because in small intestine the mechanism of action or the favorable environment of these enzymes to act is alkaline pH. So, pH should be high. So, in our oral cavity, and especially the enzymes of small intestine, they need alkaline pH, and intestinal juice has got alkaline pH. So, then the enzyme now will become active, they will convert the polysaccharide into whatever was digested from mouth, they will pass through stomach, they will reach the small intestine, they will be digested or converted to small disaccharides, and then the disaccharide digesting enzymes are present in the brush bordered cells, they will be converted to monosaccharide, for example, glucose and fructose, and then they will be absorbed through the intestinal brush bordered cells. Do not worry, we will be highlighting, we will be magnifying this whole thing very soon. I will be seeing what happens after they are digested. So, this is a chart, for example, this is a chart which shows what are the essential enzymes that can act on the foods and what is the location and what are the ultimate end products. You can get multiple choice question from each and every slide, okay, each and every point. So, be very careful that you do remember each and every one of them, because remembering them will not only help you in the exam, that is one of the goals, but it will help you to understand how you formulate a diet, because diet planning is also one advantage, as I told you, of doing this course, being a nutritionist. Okay. So, again these are the two charts. So, this is the first table and this is the second table where I have shown how all the enzymes act on the substrate and what is the location of action and what are the end product. Mind it, for dietary fibers like cellulose, hemicellulose and plant polysaccharide, there is no digestion. So, what do? They actually contribute to the stool, the bowel movement, increase the bulk of stool, and improves constipation, and that are actually fermented by intestinal bacteria. So, a lot of dietary fibers can actually lead to formation of gas, passage of frequent gas bouts, that is called flatulence. Okay. In here there lies a applied clinical importance. There is an enzyme lactase. Okay. Did you overlook it? No, it is very important. So, lactase, what does it do? It breaks down lactose into glucose and galactose. Lactose is a disaccharide consisting of 1 unit of glucose and 1 unit of galactose, and lactase is present in the brush bordered cell. If lactase is absent, lactose cannot be digested, and this is known as lactose intolerance. What is the symptom? Lactose generally is milk, okay, milk product. So, whenever an individual who is affected with lactose intolerance takes any milk or dairy product, instant diarrhea happens, flatulence, there is projection of gas, there is bloating in the stomach, there is watery diarrhea. Why? Because the thing cannot be digested, right, and it contributes to the undigested product, it contributes to the dietary fiber that will get fermented intestinally, that will increase the osmotic tension, that will lead to diarrhea. So, these are the reasons why if something is excess undigested, it will lead to diarrhea and flatulence, and in this case when it is digested for everyone, in person or in patients with lactose intolerance where there is lactase deficiency, this will be a problem. What is the treatment? It is very easy. We can simply use specialized formula, or you can simply remove all milk products from diet. Okay. Now, this lactase deficiency may be primary or secondary, means this lactose intolerance can be congenital from birth, because these enzymes are produced by genes that are developed in utero. Okay. So, if a baby is born with lactose intolerance, what will happen? It cannot thrive on mother's milk. There will be instant vomiting, baby will cry intractably, there will be swelling of abdomen, and that is the very first sign of lactose intolerance, in which clinicians and especially junior doctors who are attending the patient on bedside, or even family members who are having a newborn baby, should be aware. If a baby is rejecting breast milk, excess swelling of abdomen is happening, excess crying, might be a case of lactose intolerance. Or it can be secondary, where the lactase deficiency is acquired. Why? Basically, we have seen, it has been found that the lactase enzyme activity decreases with age. So, as we age, the ability to digest milk product is lost. So, it can or cannot happen to anybody, right. Mind it, any phenomena of aging is individual specific. You can say, okay, my grandmom always has milk in diet and roti, chapati. It's fine. She is blessed with a good lactase enzyme. Someone else might not be. Someone else might develop lactose intolerance in early 50s or 60 years of age. It varies, right? So, acquired lactose intolerance can be treated. How? We need to provide lactase from outside. Who can provide lactase from outside? By tweaking the diet. Okay, advise him or her not to take raw milk. Okay, just advise him to take curd. We can add yeast also, because these are very good sources of lactase. Why? Because it comes from the lactobacillus that is present in the curd or yeast. Okay, so curd contains lactobacillus that will provide lactase. It will help in easy digestion of the milk. Also, yeast can be used in the treatment because it is available in sachet. It can be prescribed. If present in the gut, it will provide the essential lactase enzyme. So, this alactasia will be cured. Okay. So, mind it, there is a term known as hypolactasia. Hypo means less. So, this lactase enzyme might be totally absent. It is as alactasia, or it may be less. Okay. So, it may develop with age, and this is known as hypolactasia. So, now after it is digested, we are now concerned with absorption of carbohydrate. Mind it, when we come to absorption, only monosaccharides are absorbed. Excuse me. So, what are monosaccharides? Monosaccharides are compounds that are single unit of, that are considered to be single carbon unit, for example glucose, fructose, mannose, galactose, etc. And again, for an MCQ purpose, the rate of absorption of galactose is the most, followed by the rate of absorption of glucose and then fructose. So what if the final disaccharide is not digested to monosaccharide? Of course they will not be absorbed, minute quantities may be absorbed, but ultimately everything is eliminated through the kidneys because they are water soluble. Okay. So next we come to the absorption of glucose, because this is most important. Glucose is the fundamental unit of carbohydrate. All other catabolic pathway in carbohydrate metabolism will lead to production of glucose. Also all biosynthetic pathways start with glucose, right? So we need glucose as a currency, of a ready-made currency to form higher storage form of carbohydrate. So it is very important that we get glucose absorbed through our gut, right? So the importance of glucose lies in carbohydrate metabolism, and now we should focus on glucose absorption. Now mind it, our cell membrane, since it is a protein lipid protein bilayer membrane, unless the substance is lipid soluble, it cannot freely diffuse, and glucose is no exception. So since glucose is polar, it cannot diffuse through the lipid bilayer membrane. So we need someone to handhold glucose so that it can pass through the closed gates of the cell membrane. There are few transmembrane proteins that act as glucose transporters, and via various mechanism they actually help in carrying glucose across the brush bordered cell. One example is sodium dependent glucose transporter. This is a co-transporter. What do you mean by co-transporter? Co-transporter is someone or some form of gate in which, suppose I am waiting at the gate won't open. Only if my parent is also at the gate, then only the gatekeeper will feel safe to open the gate and both of us can go in. Later on, after I am inside school, my parent can easily sneak out of the back door. This exactly what happens in the sodium dependent glucose co-transporter, right? So S-GLUT, it can also be termed as SGLT. Okay. So what it does, glucose alone not allowed. Only if when sodium is there, both sodium and glucose attached to this transporter, then the transporter simply turns the direction. You have seen revolving doors when going into the shopping mall, right? Once you are in the outside, when the whole structure revolves, you go inside, right? The whole gateway works in the same way. So once glucose is inside, our job is done. The excess sodium is also inside. It is later then pumped out with the help of sodium potassium ATPase pump. This is very important pump which uses energy. So considering the whole phenomena, when a sodium is going inside and later it is pumped out, where energy is required, energy is used indirectly. But considering the only sodium dependent glucose transporter S-GLUT, energy is not required. Okay. Now there are two types of glucose transporters, sodium dependent glucose transporter, type 1 that is present in the intestine and type 2 is present in the kidneys. Type 1 is very important, especially for, see, both are important, but type 1 is more important. Why? Because type 1 has led to the discovery of oral rehydration fluid. It is one of the very few miraculous discovery that has not been awarded a Nobel Prize. Very ironical, but it has saved countless lives. In diarrhea, for example in cholera, there are loss of electrolytes and the ultimately dyselectrolytemia leads to death. So since we are talking about glucose transporters, this is the applied importance where glucose is actually used for sodium transport. So only if we drink sodium chloride, it will not be absorbed. But if glucose is combined with sodium chloride in the solution, then it will act as wonder drug. Glucose and sodium can get co-transported, and this prevents all the dyselectrolytemia, and this is the principle of ORS, or oral rehydration salt, oral rehydration fluid, and this is the mainstay of any form of diarrhea. Mind it. So next time, due to any gastric upset, if some of us are having constant watery diarrhea, the only and only saving grace that will prevent loss of consciousness is oral rehydration salt, and that acts on the principle of sodium dependent glucose transporter 1. Sodium dependent glucose transporter 2 is also is present in kidney, and it is implicated in some form of renal cause of diabetes, specifically glycosuria, but that is not much important compared to the biological value of sodium dependent glucose transporter 1 and its role in oral rehydration salt. Extremely important, and it is a life saving emergency, right? Next, so that was sodium dependent. So is there no way in which glucose can enter the system without its parent? Of course there are. Okay, there are gates which will allow me to go to any party or club, right? So the GLUT, that is glucose transporter, acts in same way. So what it does, so glucose transporter, so this is an illustration of glucose transporter that is present, that is GLUT2. It is present in bloodstream from where glucose can be absorbed in the periphery. So glucose in the intestinal cells, it actually binds to the site of the glucose transporter. Okay, what happens? Just like SGLT, the mechanism is similar. The whole thing flip flops into the inside the cell, and ultimately the transporter, when the transporter changes its direction, glucose can be transferred inside. Okay, and this thing is reversible depending on the need of glucose. The glucose can go in and out through this transporter, whether, I mean, from what? So suppose from side A to side B, we need glucose absorption. Next, when the site A needs more glucose, glucose can go out in a similar fashion. So considering the whole mechanism, we have now magnified the intestinal brush bordered cell. So what is happening? So in the brush bordered cell, sodium dependent glucose transporter, now you know it is as GLUT1. As GLUT1 is facilitating the entry of glucose into the luminal brush bordered cell with the help of sodium. Okay, this sodium is pumped out of the cell by sodium potassium ATPase pump, because the concentration of sodium and potassium inside and outside the cell is fixed. Okay, and this is regulated by sodium potassium ATPase pump, which uses energy. And from the basolateral surface, so here are the blood vessels. So these are the blood vessels. So intestine has got a lot of blood vessels. Whatever we eat, it is ultimately absorbed in the blood. So here the blood vessel, in the blood vessel, the interface of the brush bordered cell and the blood vessel, that is the blood capillary, GLUT2 is present, which is actually capturing the glucose, and thus glucose can be absorbed via diet. So this was the enteric, this is the mouth. Right, very bad lip. So we took down the glucose, we chewed it, we digested from oral cavity, pancreas, ultimately it has entered the brush bordered cells, it has gone via sodium dependent glucose transporter 1 via GLUT2 into the bloodstream, right. So this GLUT2, the activity of GLUT2 is actually facilitated when in well fed state. Okay. So in well fed state, so see, there is a type of circulation that is known as portal circulation. There, if you are well versed in anatomy, the blood flow happens in reverse direction compared to the enteric blood vessel. So just as a rule, mind it, GLUT2 transports glucose into the cell when blood glucose is high, right. Mind it, here what we are seeing GLUT2 is transporting the blood glucose outside the cell. We are hungry, we need glucose, so glucose is getting absorbed in the blood, right. But when we are in well fed state, okay, now glucose can be stored. So this GLUT2, the phenomena will be reversed, but this phenomenon is not reversed in intestinal cells. Please do not think that when we have excess blood glucose, GLUT2 will reverse the flow of glucose into the intestinal cells, and then the whole glucose will be passed in the urine. It does not happen that, or stool, it does not happen in that way, because glucose actually cannot go out from this side. If that was the scenario, a lot of glucose would be trapped. So when we are considering inflow of glucose from blood to the cell, we are talking about liver cells, right. So, in liver, the peripheral the glucose is being delivered to the cell, where it can be taken up and stored as glycogen, and this mechanism also helps to monitor the pancreas to regulate the level of insulin secretion, because that is also another trigger. The level of glucose, whether the pancreas needs to secrete more insulin or whether it needs to check on the secretion of insulin, is a very important marker or very important phenomenon in regulation of blood glucose, which we will also be discussing later. Next, this is an example of another glucose transporter, that is Glute4. Okay. So, in this type of transporter, what happens, this is like a flip flop mechanism, where first the glucose will be captured and the mouth is opening outside the cell, the glucose molecule is getting internalized at some point of time, and then the mouth is opening at the inner side, where glucose is entering into the cell. So, this type of cell is actually present in skeletal muscle, heart, adipose tissue, etc., right. Very important, because during exercise, muscle actually accounts for 80% of total body's glucose utilization. You must have seen all the, if you have been to any aerobic exercise or gym or any yoga class or any hard work training center, for example, energy drinks are provided that have got a lot of ready glucose, because we need more and more glucose in muscle to increase those reps, so that we can build our muscle, right. But at basal metabolic rate, brain utilizes 60% of glucose. So, mind it, when we are not actively working, basal metabolic rate means during sleep actually, when nothing else is working, the brain is the main source of glucose utilization, and during exercise, muscle is the main source of glucose utilization. Okay. So, what is so special about Glute4 compared to all other glucose transporter? Glute4 is under control of insulin, whereas all other glucose transporters are not. This is again a very important multiple choice question, very important concept in understanding how insulin can regulate blood glucose, because it induces intracellular Glute4 molecule to move the cell across the transmembrane and thus increase glucose uptake. So, when there is more insulin, the peripheral blood glucose will drastically reduce, because all the glucose will get inside the cell, and this thing is hampered in diabetes mellitus, especially type 2, where there is insulin resistance, that is seen in muscle and fat cells, where mainly Glute4 are present. Insulin cannot act on them, therefore cells cannot get inside, and the level of blood glucose is very high, and it leads to diabetes mellitus, which we will be again discussing in great details later. So, these are the overview of 3 types of glucose transporter: sodium dependent glucose transporter, Glute2 at vascular site, and Glute4 in transport cell of heart and skeletal muscle. And this is a chart again for MCQ, where all the types of glucose transporters, where they are located, have been tabulated. As I told you, brain utilizes basally 60% of glucose. What type of glucose transporters? Glute3, right. So, this is an MCQ question. So, this is one example, and mind it, sodium dependent glucose transporter type 1 is present in intestine and type 2 is present in kidney, and they help in co-transporter. See, fructose transporter, who does? Glute5. So, these are very small hidden MCQs that are present in the slide. So, you should pause them, memorize, make your own notes. Okay. So, to conclude, this lecture session has covered mechanism of digestion of carbohydrate with various types of amylases in various areas of digestive tract. We have also covered how glucose transporter help in absorption of glucose across different cellular environment. So, that is it for today. These are my references, and thank you for your attention.